Okay, next we're going to hear from Lorenz remotely, I perceive he's presenting. So yeah, take it away.
Okay, hello. I hope everyone hears me loud and clear.
Good.
Hello, yeah? Okay, perfect, then I'll start. So my name is Lorenz Rumberger. I was a visiting researcher, like graduate student in Mike Angelo's lab, and I worked on a project together with Noah Greenwald and Cameron Pochara, and Dagmar Kainmueller is my PI here in Berlin. And the project is called Nimbus, and it's a tool for improving cell phenotyping by factoring in spatial marker expression patterns. So what's the status quo in cell phenotyping right now? As you heard in the last few talks, that you take like an image with a nuclei channel and like several membrane channels, and then you apply a cell segmentation algorithm. Then you take, for example, a marker that you're actually interested in, like the CD8 marker, and calculate for the segments that you have the integrated expression, meaning the like mean per cell expression profiles of these cells. And then when it's a small scale experiment, you can just like apply a threshold and say the like cells with high expression levels, they are positive for the markers, and the cells with low expression levels are negative for the markers. And this is not really scalable. That's why there are things like the cell clustering, where you can scale this over like millions of cells. So why is this cell phenotyping in multiplex images so difficult? You sometimes have abnormally shaped or like oddly shaped cells, like dendritic cells, where it's where the segmentation is like highly dependent on the sectioning plane. When you have like a nice section, a cross section of the dendritic cell, you have some like nice marker patterns. But when you have like an unfortunate sectioning plane, you have like a marker staining pattern that looks like this. And when you look at the according cell segmentation, you see that it does not correspond to any of the cells here. It's kind of like in between because it's kind of like an artifact of the sectioning plane. And you can also get overlapping signals from neighboring cells. Here's the same example that Candace showed you before with the tryptase that kind of like bled into nearby cells and thereby increasing their integrated expression of this marker, which confounds the cell phenotyping process. And what we are then agreed to do is that we try to improve upon this by leveraging large data sets and deep learning. And we want to use deep learning to factor in the spatial marker expression patterns. But what does it mean, like spatial marker expression patterns? I have here an example. On the left, you see a cell that shows the typical expression pattern of a membrane marker. As you see here, the marker closely like tracks the cell membrane or the boundary of the segmentation that is here shown in green. And here on the left hand, on the right hand side, you see another cell, but the marker doesn't really track the membrane here. It's like kind of like probably from a nearby cell or some other reasons why this marker is here. And when you look visually at it, you clearly see that there's a difference. And hopefully deep learning does not have any problems with like discerning these two. But when you just average the expression levels for the segments, you end up with like kind of like the same expression levels for these two cells. So why again deep learning? You heard today that you can reach human level performance in whole cell segmentation, given you have like a large and diverse data set. And there's publications where people reached like physician level performance in melanoma classification, or even superhuman performance, meaning better than any individual human physician in breast cancer screening on mammography. But all of these have in common that the performance deteriorates if the samples that you want to apply the algorithms on are like really unlike any samples in your training data set. That's why we composed a really large and diverse data set spanning over 61 different protein markers imaged with different modalities like Vectra and Codex, which are like multiplexed IF platforms, and the Mibiscope, famously used in Mike's lab. And with different tissue types like colon tissue, decidual tissue, pancreatic tissue, and tissue from triple negative breast cancer. And also these all these samples were acquired, all of these sub data sets were acquired by different labs, some of them at the Memorial Sloan Kettering Cancer Center, and some of them also at Stanford University. So what is the idea again? So when we look at these two cells here and we look at their integrated expression, it's really close to one another. And when you look at the like KDE plots of integrated expressions, they often look like this. So it's like an unimodal distribution, and it's skewed, and you cannot really say where should I like make a distinction between a positive and a negative cells. And these two different like cells, even though their staining patterns really distinct, you lose information when you integrate the expression. But with our solution, which is in essence, it's a data transformation, so that the spatial marker expression patterns, like the different spatial marker expression patterns, are accounted for and factored in. And when you use Nimbus to predict if like a cell is positive or not for a given marker, this transformed per cell expression becomes like bimodal distribution. And it's really easy to say, well, there should be some cutoff over here where you can say the cells on the left-hand side are negative and the cells on the right-hand side are positive in this KDE plot. So where does it fit into the pipeline? You heard that after like normalization, the first step is the cell segmentation step. And then you can take the cell segmentation mask and the marker channels, apply the Nimbus marker confidence scoring, and then you do cell clustering based on the Nimbus predicted confidence scores with, for example, Pixie or other tools. And how did we exactly do it? So we constructed a really large data set, and we leveraged publicly available data sets mostly, but also some data sets that are not yet published. And we first created some silver standard labels. So these publicly available data sets, they come with the cell table, which maps like individual cells to cell types. And then they come with a cell segmentation mask and with the individual marker channels. And what we did is we looked into the cell tables, and they were mapping like cell types to individual segments. And for example, when we have like a cell type A, we know this cell is of cell type A, we map back from the cell types to the marker expressions. So for example, marker A is specific for cell type A. So we could generate like a map that says this cell should be positive for this marker, and these other two cells should be negative for this marker. And the same could be done for marker B, which like the cell type B should be positive for marker B, and cell type A should be negative for this. And this is how we constructed kind of like automatically our silver standard labels. And in order to validate our tool, we created the gold standard labels. And you know how gold standard labels are created. You like get a bunch of expert annotators, which mostly means graduate students, and then they will just like look at every individual channel at the individual cells there and tell you, is this channel, like, is this cell in this channel positive, yes or no? And here's a small summary of our training data set so that you see the scale of the operation. On the y-axis, you have the number of annotations in 100 millions. And here I split the data set by the imaging platforms into MIBITOF, Vectra, and Codex. And you see we have like 95 million MIBITOF annotations in our data set and around 50 Vectra and Codex annotations. Here you see the annotations per tissue type, again in 100 millions. And you also see that the most of the annotations were from a large breast cancer data set, and we have two different colon cancer data sets. Here's the number of annotations per marker location. So we bind the markers into membrane markers, extracellular matrix markers, and nuclear markers. And membrane markers far outnumbered all of the other markers. And here we have the number of cells per lineage. And there we just bind the cell types assigned by the authors of the original studies into lineages. And you see that most of the cells ended up being in the other category, or epithelial or lymphocyte cells. And what we proofread is it just accounts for 0.6% of the total number of annotations, but we proofread more than 1 million cell annotations. So even though this sounds small, this is a pretty large number. And this means like 90,000 cells on average in 11 channels for all the data sets. And I compared this to like all the previously published like gold standard ground truth annotations in multiplex data about like if a cell is positive or not for a given marker, and it dwarfs the other data sets. It's like 15 times more manual annotations than all the previously published studies combined. Okay, and since we have like the gold standard labels, we can do quality control on the automatically generated silver standard labels. And we calculated the precision and recall and the harmonic mean of the two, the F1 score, and ended up with like around a 0.75 for the silver standard compared to the gold standard. So what does our model do? What goes in, what goes out, what's the structure? We use a UNet as the backbone network, which is like the workhorse of medical computer vision. And the input is a single marker channel and a binarized segmentation map. And the network learns to predict pixel-wise marker confidence scores that tell you if a cell is positive for a given marker, yes or no. And we used a noise-robust training procedure to leverage the silver standard data set and a silver standard labeled annotations. So does it work? That's the question, right? And here I averaged the Nimbus confidence scores and the integrated expression. So what you see on the y-axis is the log density, and this is like a kernel density estimate plot of the Nimbus confidence scores on the top and the integrated expression at the bottom. And I colored the plot by the ground truth negative and positive cell types. And you see that indeed, like with the Nimbus confidence scores, you get a nice bimodal distribution, whereas with the integrated expression, it's kind of like metal all over the place. But let's zoom in more to understand what like the predictions really look like. So here we have the Nimbus confidence scores again on top, and the integrated expression, the histogram, like the KDE plot in the bottom for the tonic data set and for CD8 cells. And when we zoom into one like specific example, you see here again the CD8 channel, and you see that the marker like tracks the membrane of the cell. So this cell got identified by our expert annotators as a positive cell. When you look at the integrated expression level, it's quite low. It's actually 0.02. Whereas when you look at the Nimbus score, it's 0.82. So this cell that has like a really low integrated expression got picked up by our model, predicted to be a positive cell because of the distinct staining pattern. And here you see the opposite, I'd say. Here's a cell that is CD8 positive, and here the marker bled into a nearby cell. When you look at the integrated expression, you see that it's pretty high here. It's around 0.1. whereas it's on the Nimbus score, like the prediction by the model is around 0. So here it accomplished to understand that the marker just bled into the nearby cell, and it's not actually the marker expression of this cell, because it also does not like track the membrane of the cell. And here we did the same for the Hickey colon data set and the cytokeratin channel. And here you see like cytokeratin is a epithelial cell marker. Epithelial cells are densely packed and there's lots of like marker expression all around here. But when you look at the integrated expression of this cell, you see it's again like really like low. But when you look at the Nimbus score, the model was able to learn that like bunch of like marker expression all around here means that this cell is most likely positive for this marker. And here we have another example of the opposite, a nearby cell that got identified by our annotators as negative. And again, there's like marker expression bleeding into the cell. On the integrated expression, it looks like it's positive, whereas on the Nimbus score, it looks like it's negative. And indeed, our annotators agreed with this. So we evaluated the model versus the gold standard labels. And then like the F1 score precision recall is around like 0.8. And then it's higher than the silver standard labels that the model got trained on. And then we looked at the individual data sets, and there it's the same picture. So like when you look at the silver standard labels, they are worse than our Nimbus predictions for every individual data set. So our noise robust training procedure actually improved the model, even like the silver standard labels are not perfect. So here we split the performance by marker localization. It works well for membrane, ECM, and nuclear markers. Here you have the F1 score split by tissue type, and you see it works well for breast, pancreas, colon tissues. A little bit like less so for this decidual. There the F1 score is around 0.7. I think it's like the data set is more noisy, and also it has more like segmentation problems because the cells in the decidua haven't really, like they have really unusual shapes, and there are more like false merges in this data set than in the other two. And here we have the F1 score split by lineage, and it is the same picture. It works pretty well for cells no matter what the lineage is. So how can you integrate this into your own cell phenotyping workflows? The model is publicly available on GitHub, and here's the link. So if you want to try it, you can just download it, download the model weights. It's easy to run. I compiled a Jupyter notebook where you don't need to be like a computer scientist to apply this on your data. You can directly load the model checkpoint and run inference on your data. That means you don't need to like annotate your own data. You don't need to retrain the model. You can just like load the checkpoint and it should generalize also to your data. And you just need to like put in the mask and the marker images, and then you get the predicted confidence scores, which can be averaged over the cells into a cell table. And then the cell table can be plugged into like one of the many cell phenotyping platforms that people use, like Stellar or Celesta or Pixie for cell clustering. So the conclusion is cell phenotyping can be improved by factoring in the like spatial or sub-cellular marker expression patterns. These confidence scores nicely separate the data better than the integrated expression does, which I showed in like many examples, I hope. The pipeline is publicly available and can be run without any like labeling, fine tuning, anything. Also like on notebook hardware on at least small data sets. And a preprint is coming soon. So with this, I'd like to thank my collaborators from the Angelo Lab and also my collaborators in Berlin. And you for the attention, of course.
Questions?
Excellent work. Can you hear me okay?
Yeah.
So one question I had was, and maybe I missed this, but is there a way to also incorporate like known biology into the model? You know, you would expect CD3 and CD8 to overlap, for example, and that might help improve the confidence score. And then the second thing is kind of in alignment with what Candice talked about in terms of the scalability, in terms of the number of cells or images. What can you give me a sense of how long it takes to run for a data set?
Yeah. Okay, so the first question was about the marker co-expression patterns. Like I think you said CD4 and CD45 or CD8 and CD45 or something like this. And at the moment, it cannot be incorporated in the model. You get like for every single marker, you get a prediction if it's positive or not. And then you can use like cell clustering tools that leverage the marker co-expression patterns like Pixie, for example. And we hope that by this you can like factor in this like biological prior knowledge or this like co-expression patterns that you know of. And the runtime of the model on my laptop GPU, it takes I think I can do inference on 4 samples sized 1024 pixels squared in one step, which is like a second. Yeah.
Thanks for a very clear presentation. Looks like a great approach. I'm curious, there are biological contexts where molecules get polarized to one face of a membrane for a reason, like say cell migration or an immunological synapse. Does that information get lost using this method, or have you guys thought about ways to try to retain that?
That's a really good question. So I think like since this method is at the moment work in progress, I think this is like a feedback where I would say I need to check in our predictions what happens in this case. So when like a marker expression gets polarized in specific cells. So I don't know yet, and I need to say I haven't thought about it yet. But thanks for bringing this up.
Mike, that's a really, really good point. And I think scoring stuff like immune synapse formation or those types of asymmetries is something that is going to be really important. My suspicion is based on the training data that we have, is that it's going to miss it, I would guess. And so it's you know, because kind of what we went into, you know, I mean, Noah and Lorenz like really just like drove this whole thing, and I think like sort of conceptually the goal was to have something that essentially factors in sort of human level intuition with looking at expression patterns around cells. One, and then two, to have something that was kind of panel agnostic, so that you know you could use this on anything without having to worry about if your panel fits sort of, you know, bespoke things for certain cell types. But I think it definitely was sort of envisioned and sort of factoring in things about conventional cell staining into the probabilities. And so I think things like, I would guess that stuff like, I don't know, I mean, it would be interesting to think. I think the problem is we don't have any, we need to get good ground truth data on that stuff so that we can test it. But yeah, I wouldn't be surprised if we didn't perform as well on that. Any other in-person questions? No? Did you see some in here you thought were good? Yeah, so this one, go ahead, Lorenz.
I just wanted to say I can answer the online questions online if you want, but you can also.
Yeah, I think in the interest of time, we can move forward, but then, yeah, you're a panelist, right? So you can just type on there,
yeah.
Lorenz?
Yeah.
Okay, perfect.
Yeah, I can. Okay, then thanks.
Thanks, Lorenz.
Next up, we have Yuval from the Weizmann Institute, who's going to talk to us about their new cell classification pipeline.
Hello, everyone. My name is Yuval. I'm a PhD student at the Weizmann Institute in Leeat Keren's lab, past postdoc from the Angelo Lab. And I'm really excited to share with you another method that a solution to the same problem called CellTune, a human in the loop framework for efficient and accurate cell classification. And I'll say that a lot of the challenges that have come up in the past few presentations are the exact same challenges that we have. So I'll go through those. I'll skip some of those things, and I actually think that a lot of what we've been working on is synergistic. So it'll be really nice to see how things progress in the next year. Okay, so we've already gone over this, but our typical computational analysis pipeline and where cell classification fits in after segmentation. And there's all kinds of tools that have been used. And clustering is just kind of the prevalent one that we show in these examples. But really the importance of this is that all of our downstream analysis depends on having accurate classifications. We don't actually have a quantification yet of how good does your accuracy need to be to get to the relevant question that you're asking to be able to accurately give the answer to that. But what we're trying to achieve is at least get as close to human level as possible with these things. And we've already seen that there's this overlapping spillover from neighbors, projections from different cells. And of course, we always have some noise and still some segmentation errors that lead us to get these clusters of cells that have expression of multiple different types of lineage proteins. And so there's been all these works, trying to deal with this in different ways. But basically, these all funnel at the end to some step which either involves clustering or gating at the end, like with Pixie and Nimbus. At some point, you have to get to this clustering or removing the spatial expression information or doing a lot of manual labeling and curation. So our first question was, can we utilize the spatial expression information and how should we do that? And our first try was to use a CNN, and this is called CellCider, and it's published in Nature.com this year. And here we just had manually labeled a lot of cells and used CNN an ensemble of CNNs to classify these with the confidence. But what we learned is that this really requires a lot of manually labeled training data, which, as everybody's repeated, we don't have a lot of that, and it takes a lot of effort. And with this, we had kind of a very limited interpretability at the end of what went wrong in the cases that went wrong, and why does it make certain classifications. And so this brings us to this question of what's the most efficient way to learn. And there's all these linear approaches, and you take the cell table, you do some clustering, you get the clustering, then you have to label the clusters. You have to identify what cell types you have, then you do some probabilistic model, you get predictions, or you manually label cells and then use some supervised learning. But all of those ended up having manual curation built into them that's kind of hidden, but takes months and months of work. And as Noah said in his talk, human in the loop active learning really is an efficient way to learn, and it works well for segmentation. So here we use it for classification. And the principles that guided us is to use those spatial features to learn from human knowledge and be able to input human knowledge into this and to minimize our labeling efforts. So clustering and gating doesn't do the first two, but minimizes the labeling efforts. The CNN doesn't minimize the labeling efforts. And hopefully I'll show that this does all three. So the workflow, we have to start with some labels and some features for each cell, just like with the cell table, and then do a training and prediction. And I'll talk about each one in depth. And then as in any active learning framework, you need to measure uncertainty somehow so that you can, and sample cells so that you can give those cells for manual labeling. and then add those and continue. And there's this other step of initialization. How do I get some kind of starting point of labels and features to get this rolling? So for the initialization, what we did is we extracted features, spatial features from each cell. So we took the cell segmentation that we got from Mesmer, we expanded it a few pixels, we eroded it, made it, shrink it some pixels, and then we also looked at the environment, and then we calculated for each of those different segmentations, the expression of each marker in there. And then we looked at, for each cell, its neighbors, and what was the expression, the maximum expression of a neighbor. And this helped us to capture these spillover effects where we had really high expression of a neighbor and a low expression on the self. And it's flexible to kind of use anything you want to put into it. So we've had a lot of people adding in regional masks, which they quickly train in Elastic. Takes them about, a day. There might be something that they're interested in. So for example, if you have smooth muscle vascular structures and you want to differentiate those from the fibroblasts, both are expressing SMA, you train this quick pixel classifier to identify the vessels, and in that way you add those. And you could add in, and once you do this, then you extract for each cell the values of the proteins in the cell, but also in those spatial domains and the neighbors and region masks. And it's really flexible for any input. You could give any kind of pixel classification like Pixie as an input. You can give the probabilities like from Nimbus as an input as well, and cellular features like morphology, things like that. But how do we initialize the labels? That's the next question. So we were inspired by Spitzer 2015 science paper to do landmarking, which is really just a simple high threshold where you're super positive, and this ends up being around 10% of the cells. You could think of like in the case of Nimbus where you just say, okay, everything above 95% probability for this marker and not high on the other markers would be the landmarks. And here's like just an example of a macrophage which has super high CD68 and doesn't really have much of anything else on it. So we get those as our initial step in that, and that doesn't take us very long to do automatically. And then we have our labels and features. We train and predict with gradient boosted decision tree models like XGBoost and CapBoost because they are very fast. They're very good at learning from tabular data when you don't have tons of labels. So what we do is we take actually two different models and we get the predictions from each one and we compare them to each other. And in this way we can measure uncertainty of where are the cells that we should be labeling. And this is a well-known tactic called query by committee, where you take the disagreement among an ensemble to select data for labeling. So here we're just taking two models and we're selecting the cells where they disagree. But still, that's a lot of cells when you start out. And so how should we sample the specific cells that we're actually going to label? Because we don't want to label thousands of cells. So there's many different ways to do this sampling strategy. And what we do by default is balancing agreement across fields of view. So you get a subset of your cells to do that. Balance cell type agreement, so across different types of cells. Prioritize rare cells, which we are often missing and we're often getting low F1 scores for. And then the user can also input any cell type or confusion between cell types that might be interesting for them. So if you're doing a project that's asking a question about macrophages and you really want those to be super accurate, you can prioritize those for your labeling. And then you get to the manual intervention part where a person has to label the cells. And this is not trivial. And our question was, can we also make this process faster? So we teamed up with the Parker Institute, and specifically Rob from there, which developed the Mantis viewer. And we added a bunch of features to enable you to label cells very efficiently. And so now when you hover over a cell, you have a cell legend, which will show you a sorted list of the expression of the markers in that cell. And when you're doing this with CellTune, then you have inputted also in the legend the two different types of predictions. So this cell in the beginning was predicted to maybe be either a cytotoxic or a T helper cell, and there is some CD4 expression on that cell. But when we look at it by eye, we see that it's very, very clear to us a CD8 expressing cell, and there's some spillover from the neighbor. And so you have this population panel where it's easy to just click and adjust the populations for that cell. And that's one thing we've done. And then the second thing is we've tiled and ordered the cells so that the same confusions come up in a row. So you're just going through a bunch of CD8, CD4 confusions at first, then you switch to CD4, T macrophage, etc. And this way we minimize the amount of time you spend changing the different proteins which are visible, since we can only see 7 at a time. And lastly, we added channel mappings, which are just user-defined groups of markers that can come up all at once by just selecting them. So this saves us a lot of time in setting the channels. So a lot of times, if you're just starting from scratch, let's say you're not using CellTune, you just want to label some new data set. You have an overview group, which gives you an idea, is it an immune cell? Is it a tumor cell? Is it an endothelial cell? And then you say, oh, it's an immune cell here. So you have an immune panel that kind of narrows the space down. So with like 3 clicks, we're able to quickly see this is, oh, it's a CD8 T cell. And this is an example of what it looked. So those are schematics. This is what it looks like actually with the tiled cells. And you have a zoom in on a specific cell where the user labels the center cell. They adjust the label here. You have 2 predictions, and then easy to navigate to the next tile. So the principles, those 3 principles about spatial features, learning from human knowledge, minimizing labeling efforts, how we've implemented them, extracting these features per marker, training pixel classifiers, which is optional. You can also add in things like Pixie, using supervised machine learning, human in the loop, like what Noah did, and then having this, prioritization for like a specific thing that you're interested in. And all these features that we added to Mantis really speed things up. So results, we had a manual labeler sit and label 3,000 cells recently, and we saw that the agreement between the predictions from a different person using CellTune was 85%. The x-axis is the CellTune predictions type, and the y-axis is the manual labeler. And so we have calculated recall and precision for the different cell types. But then we said, okay, but some of these are ambiguous, and some of them maybe CellTune's doing better than the person. So we took the cells off the diagonal. These are where the manual labeler 1 disagreed with CellTune, and had a different manual labeler look at those cells and decide, is this who's correct, or neither, or both if it's ambiguous. And here we actually saw that the disagreement analysis favored CellTune, and that 44% of those cells, the second manual labeler expert agreed with CellTune, whereas only 19% with manual. And then we had 3 manual labelers where we took the majority vote to be this consensus for these cells. And we saw that the CellTune predictions were slightly better than the manual labeler, but a jump above what we were getting with other methods. We tried different types of clustering, giving the optimal gating, knowing what the cell types were, and then we only get to 80% agreement. And when we look at the F1 scores, it's more drastic because you have these rare cell types which have low F1 scores. And so we can look at the actual cell types for this example, and you have like mast cells where the cell clustering was doing a really poor job, F1 score of 0.5, and CellTune was right there, 0.95 with the majority vote. And there are other cell types that have nuclear staining markers, which are easier, like Tregs. But overall, we're really happy with what we're getting, and we found that it's learning quite efficiently compared to just labeling, fields of view or labeling random cells. As we label cells, our accuracy increases faster. We've labeled 5 data sets with CellTune. It's totaling over 1.5 million cells, where we're running this process until the disagreements are really ambiguous. So the user just like stops, and then we have now these data sets which we're very confident in the labels, and this will be hopefully a good resource for the further development of more deep learning approaches and things like that. So just an example data set from lung cancer. You can see on the right all of the cell types that were in the end added along this process, and it's really a lot more than what we've been getting with other methods, and even in regions where there's a lot of B cells and T cells mixed together, we're getting nice representation of the different cell types. And one of the things that we really found useful is that as we're doing this, it's facilitating us finding the unanticipated cell types because we're interacting with the data. So for example, CD8 Tregs were not in the list of initial cell types, but they come up really fast as a confusion between CD8 Ts and Tregs, expressing both FOXP3 and CD8. And if she didn't put an NKT in this example, so that came up too. And then plasma cells, which had CD31, and type 2 pneumocytes, which express both DC LAMP and keratin. So, and then lastly, it's really highly interpretable since we're using these XGBoost models and they're very fast, and we can see what's the model actually learning. And for CD8Ts, it's obvious it's going to learn features to deal with CD8 in the segmentation, which is road 0. But also it learns, to look at the neighbor's expression, and if the neighbor has high expression, to lower the probability of that cell being a CD8. And here we have for Tregs, FOXP3, now it says a road 5, so it's looking at the segmentation which is closer to the nucleus. So in summary, we've achieved human level accuracy for this cell type classification. It's been a fraction of the time in that it takes us only each person around 2 to 3 weeks, whereas before we were spending about 6 months per project. The labeling features that we added to Mantis really improved the efficiency of doing this labeling. And the process is really more efficient than labeling the whole fields of view. It facilitates the cell type discovery and is interpretable. And it's a really flexible framework in that you can customize it as you want to find rare cell type specific confusions. You can continue until you're satisfied with accuracy, and you can also use other methods as an input. So thank you, and thanks to my lab, Leeat Keren, and Dana, who's working with me, a master's student, Sarah, who labeled cells. And this is coming soon as both preprint and code, but look out for it on Leeat's Twitter. Thanks.
This looks really exciting because I really like the fact that you can put in, virtually any kind of parameter for the cell classification. So one of the questions I had is, so do you think, because I'm just sitting here thinking, it's like, we could put in like pixel clustering frequencies, we could put in like, Nimbus, and is for the algorithm, will it prioritize which parameters to pay attention to? Like, I mean, do you run the risk if we did something like that? Would you run the risk of sort of, putting too many parameters into where it would have trouble sort of honing in on the right classifications, or do you think it's going to be robust to that?
So I definitely think it's going to be robust to that. Since these are tree-based models, they inherently need to do some feature selection as they build the trees. And that's actually one of the things we were going for, because with the CNN, looking at the image, there's just so many features in that image that we need tons of labels to overcome, overfitting. But with these tree-based models, even with a few thousand labels, they're able to just kind of select the best features to use.
And the only other question I had was if you so just in your experience in Leeat's lab, when you're doing this in a cohort, so you've got sort of like the threshold based high confidence for the seeds.
Yeah.
And then how many corrections do you need to do for the human in the loop usually till you feel like it's dialed in as well as you want?
Yeah, so we usually do around 3,000 to 5,000 corrections. Takes about 2 weeks.
Yeah, that's not bad at all. That's awesome.
Yeah.
How are you doing, Tan? Yep.
Sorry, I have a question. Thank you so much for that and for speeding up everything for us. But my burning question is, hopefully we can eventually do H&E staining on the same slide as a MIBI. Will there be a way to incorporate the images, the cell types you can see on H&E? Because on H&E you can see Paneth cells, plasma cells, endothelium, bronchial epithelium, cancer cells with 100% accuracy, and you can maybe put that in first as your ground truth. And then we're really bad at seeing what are T cells maybe and B cells and mast cells probably we can't see very well. Macrophages can be very confusing, but you know for sure those endothelial cells are just with muscle cells on an H&E.
Yeah, definitely. So it's definitely flexible that you could put in the H&E image. You could put in anything that you've classified before, or even the output of a classifier that you've created. So yeah, I think that's a nice way to add that information in. But the question would be, do you need to train kind of a classifier on the H&E and then input that the output of that, or just put in the H&E images and give the pixel values for each cell from the H&E? It's TBD. We haven't tried that.
Any other questions in the room? We've got some on Zoom, if not. No? One question, do you use the noised or raw images for labeling?
So we've used the denoised images, and recently, I haven't prepared slides on it, but recently we've been testing what if we just put in the noisy images, and the results are not that much worse. But we're analyzing, the effect of denoising for our upcoming preprint.
And then we've got another one here. For the annotation workflow, will it be added to the Mantis viewer main branch, or is it directly integrated into CellTune?
Again, the labeling workflow is already in Mantis, but in order to kind of import or generate these tiled cells, that's done in CellTune, and then you just open your project in Mantis and it loads it for you.
And then is it useful to have a quote unquote, I can't tell what it is category during the manual cell labeling step?
That's a great question. So I didn't mention it, but we instruct when a person's labeling to skip the cells that they don't know. That way you're not training on that cell, but it also, as it goes along, it does retain a list of the cells which were ambiguous and skipped. So we're also looking to see if we can, predict the cells that are ambiguous and kind of leave them out. But that's, yeah, but we do skip those cells.
And then one other question we have, I expect some markers to be co-expressed. For example, CD8 cytotoxic T cells would express CD3. When you indicate CD3 cells, do you mean they're CD3 cells without other subset T cell markers? What are your strategies in categorizing subsets of immune and mesenchymal cells?
Correct. So the CD3 T class is the class of T cells which don't have CD8 or CD4 or FOXP3 in our data sets. And one of the ways that we found was useful to add as a feature was to create co-localization masks, which we do automatically. It just gives, if CD8 and CD3 are in the same location, the values there. And then for those CD3 T cells, you get a high level of CD3 without CD8 and CD4. And we do the same thing for basically all of them.
Okay. Well, thank you.
Thanks.
For our next speaker, it's David Van Valen, who's visiting us from Caltech. Here he comes. I wanted so badly to play a Van Halen song while you're walking up here, and I couldn't get it together in time, so I'm sorry.
All is forgiven. Fun fact: Eddie Van Halen was a Pasadena native. Yeah, so there's a little bit of history there. Okay, so I have 19 minutes and 56 seconds to walk you through about half of our lab's research portfolio in spatial biology. One of the worst things about being a professor is that my email is no longer useful. My inbox fills up like literally every 30 seconds. And as part of that, I did not look up the amount of time that I had to speak until after I had made most of the slides. And once I saw it was 20 minutes, my response was to add one more slide because it was too, it was too cool. Okay, so let's get started. So disclosures, I'm currently chief scientist for Barrier Biosciences. They have a license for a large fraction of the software tools my lab works on. And then also some of the research results I'll talk today are currently unpublished. So just first one, take everything I say with a grain of salt. Second one, just, you know, I don't want to say like don't scoop us, but please don't. Okay. So, you know, why we're all here, just because, you know, we've had these revolutions in spatial biology and AI that are just really transforming our understanding of living systems. You know, old imaging data, 3 colors, look at patterns, you know, textures and whatnot. Modern imaging data, I like to think of these as N color images, and is now a number between 12 and over 1,000, where each color gives you some information on some biologically meaningful variable. If you're in the proteomics world, this is on the order of 12 to 40. If you're in the transcriptomics world, this can literally be several thousand. And so these new spatial data sets are interacting with the advances we've had in AI through AI making these modern spatial data interpretable, right? And we've been seeing flavors of this theme occur all morning. And, you know, what's really exciting are just all of the different biological insights and, you know, drug discovery that can happen if we're able to really access the latent information in these raw data sets at scale, right? Understanding spatial variation of gene expression across different tissues, looking at the parcelless variation within tissues while preserving the spatial information, looking at different motifs of spatial organization, and then also using the spatial information to sort of regularize the target discovery process, and then also identify spatial biomarkers for disease, both progression and then also response to therapies. All of these are things that, you know, can be ours if we make all of this data interpretable. And that's really what the AI is here to do, is to make things interpretable. This slide came from one of Aviv Rege's papers, Bobby and Tsai, so I didn't make it, no need to take pictures. You can just look it up. Okay, so, you know, if there was one thing that you take away from anything I said, because a lot of the work that I'm going to show has been talked about before, either by Noah, the cell segmentation, and also you've seen a lot of stuff on cell phenotyping. The big thing that you should just take away is that the deep learning revolution is data driven. There's no revolution without data, and in this space, it's really true that there is no revolution without labels. You need both the raw data that speaks to the space of things that you want your AI methods to work on, and you need labels that contain the insights that you're trying to get these AIs to extract. If you have both of them, then you're in business. If you have one but not the other, it's hard. And then if you don't have either, well, good luck. And so when this talk is labeled "Everything is Code," it's really just understanding that, the algorithms are great, the software systems for deployment are great, but the data itself and the underlying labels is an integral part of these software systems, and they have to be treated on the same playing field as the software code that's being written. Okay, so with that being said, a large part of what my lab is trying to do is just create these computational primitives for cellular imaging. And it's really just spanning the entire space from cellular systems in a dish all the way to cells and tissues. And really what we want to have are AI systems that can look at scenes like this. So this is a classic cell biology movie of an immune cell, neutrophil, chasing a bacterial cell, eventually finds it, engulfs it, and eats it. We want to have AI systems that can look at raw data like this, so the raw sequence of frames, and then extract a narrative of what's happening in these data sets that's similar to what a human would do, but with entity level resolution. And so you can go through a thought exercise of what this might look like. Very first notion is okay, what's a cell? Should be able to identify all the cells, and then also in this live cell imaging movie there should be some temporal consistency. So you should be able to follow the same cells over time, extract information like what they look like, where they are, et cetera, et cetera. But then there's also this, sort of semantic or biological information that's layered on top that these systems should be able to access as well. Notions like cell type, notions like cell-to-cell interactions. Right at the beginning of this movie, there's interaction between this immune cell and bacterial cell called chemotaxis. Towards the end of this movie, this interaction changes to phagocytosis. You should be able to understand that, and then you should also understand that when this phagocytosis event happens, there's a change in state from this bacterial cell being alive to being dead. And so already you can sort of see these core building blocks that this system needs to have. Finding the cells, cell segmentation, following over time, cell tracking, identifying the cell types, and then also identifying these cell-to-cell interactions. And really what these computational primitives do is take you from the space of raw imaging data, it's kind of tricky and challenging to work with, into this more interpretable space of spatial temporal graphs. The relevant entities are nodes in this graph, and then you have node level features like cell types, and then you have edge level features which denote the interactions. And so building up these computational primitives, both from the algorithms side, the deployment system side, and then also the data side, this is a core mission of my lab. Okay, so with that, I'll just walk you through some of the stories about building computational primitives. Very first one is single cell analysis. Finding single cells in movies. In our lab, we do a lot of live cell imaging in cells and cell culture, and get really gorgeous data where you have these cells expressing biosensors which report on the activity of particular signaling pathways. You can pair this with endpoint spatialomic assays, and great data. But if you want to quantify it, what you have to be able to do is find all the cells in every frame, link them together over time, and then ask, okay, in each compartment, how bright is it? And then you can go from like those movies into these quantitative traces. For the tissue imaging space, very similar problem. You have gorgeous experimental platforms which can give you these end color images, very rich data sets. That's taken, I think, one of the first, landmark papers in the space by Mike and Leeat. Great data, very rich, but you have to be able to find all the cells, right? And so, some person or some algorithm has to go through these data sets and identify, okay, these pixels belong to cell number 1, cell number 2, et cetera, et cetera. If you can do that, then all this downstream things start to become accessible. Cell phenotyping, looking for spatial patterns, tying these to outcomes and whatnot. And so really the two computational primitives here are, for the live cell imaging, it's cell segmentation and tracking. For the tissue imaging, it's both nuclear and whole cell segmentation. The path goes through, just generating data. I'm not going to belabor this point. It's complicated for the tissue imaging data, just because there's a large variation in cell morphology and signal to noise across the different platforms. For the live cell imaging data, it's just really hard because here you have this problem of time. Everything needs to be temporally consistent, both the labels and then also the labeling of dynamic events. And it's not just, and what you also have to do is label the full distribution of events, right? Cell divisions, while our textbooks tell us, one cell turns into 2. When you're looking at a lot of transformed cell lines, this isn't what happens many times. And you need your labeling framework to be able to capture these sorts of events so that you can actually have the AIs pull them out for you. And so there's the individual data challenges, each data set being quote unquote hard, but then there's also a scale challenge as well. You just need a lot of examples to get these AIs to work well. And so, what we just realized, a few years ago is that, this problem is, too hard for the AIs to do it by themselves. It's too hard for the humans to be able to do it by themselves because of the issue of scale. And so the correct solution is to have the AIs and the humans work together to build out these large data sets. We've built software systems to enable this sort of interaction. And the key thing that you should take away here is that, the way in which you get scalable labeling is by having humans correct model mistakes, because then you can take advantage of your models improving performance as your labeling process continues. And so we built this software system, Deep Cell Label, that lets us do labeling of large data sets in a distributed way. And so the key thing here is that this is browser native and also scales as your user-based scales. So if you have, one data set, a dozen, 1,000, you can still have the same sets of people. And this can be used for both crowdsourced workers who have less skill, and then also expert workers who are sort of more the PhD and potentially MD variety as well. And we have different modes for labeling, both for cell segmentation, live cell imaging data, cell phenotyping, et cetera, et cetera. So an extension of the work that Noah presented earlier is that we've actually gotten this human-in-the-loop approach to work for entire movies. And so we just submitted for publication, and we're gearing up to make the preprint available, work outlining how you can do this sort of iterative human-in-the-loop labeling for dynamic movies. Data set similar in size to the original TissueNet, but now you have this temporal consistency constraint imposed, and then also these dynamic events. And with these data set, you can create models that do both segmentation and tracking. And not surprisingly, because it's just seen many more examples, it performs much better on the cell tracking problem. Similar story for tissue imaging data. I'm not going to belabor the point. Mesmer works really well across a wide variety of different use cases. And its biggest advantage is being powered by a large, diverse data set. It doesn't contain everything, but it contains many things. And so oftentimes, this is a very good starting point. One thing that we've been working on recently is just, continuing to reduce the marginal cost annotation so that you actually can go from, most things to everything. And here we've been inspired by work released by Meta recently on a method called segment anything. And so what this method is a transformer-based instant segmentation model that's interactive. And so you have a large vision transformer that's been trained on a large set of data, and you can ask this model to generate masks with different prompts. So you might be able to say, hey, look at this point in an image. You might be able to say, hey, here's a bounding box, produce a mask. You might even just give it some text and say, for this case, it's scissors. But for our case, it might be T cells. And so this is something that I think is really, has potential to be powerful, because you could potentially take a labor-intensive task generating these single cell masks and make it so that, you just potentially point, click, and a single interaction generates a medium to high quality mask that, requires minimal refinement. The problem with this method is that, one, there's differences with the cellular and tissue imaging data that sort of prevent it from working well, and then training data sort of quarter that. And second is these vision transformers can be very expensive to train. And the third is just the way in which you ask these methods to generate masks for you. That's something that hasn't been well worked out for the nuances of these data of these cellular imaging data. But we've been working at it, and we've got versions of the segment anything model where the performance is on par with what Mesmer can do for the tissue imaging data, and it also works fairly well outside of the tissue imaging data distribution. So if you're looking at cells in cell culture, bacterial cells, and whatnot, the performance is maintained. And then also excitingly, this is very early work, but we've been working on extending this to 3D. And so many people have asked my lab, hey, Dave, when are you going to work on 3D? And my stock answer has been like, well, whenever somebody comes and drops $10 million in my lab, that's the only thing that will make it worthwhile. But if the marginal cost of creating those labels is more limited, then 3D annotation is something that we could potentially do. And we have some preliminary data showing that we can extend these models in a way where they can be prompted to generate reasonable quality three-dimensional masks. Okay, the remaining 7 minutes, just talk about what happens after cell segmentation. Here, I just want to bifurcate into the proteomic space and the transcriptomic space. Proteomic space, as everyone's seen, the big thing that's remaining is cell phenotyping. For the spatial transcriptomic space, big in my view, unmet need is spot phenotyping. And we've been working on methodologies for both of them that are both accurate, but then also generalized. And so I won't belabor the point on why cell phenotyping is hard. Right, you have all of these different artifacts, and from segmentation errors, noise, marker spillover, and whatnot. But I would argue that this is actually something that's fairly straightforward to address because it just maps you into doing an image classification problem, right? You find where the cells are, to crop out some region, create enough labels, and most CNNs will do a good enough job where this is manageable. The real challenge that's remaining is generalization, right? And the reality is that the space of different spatial proteomic methods is quite vast. You have the mass spectrometry-based methods, which are really nice. You have fluorescence-based methods that are really nice. They all differ, and the nuances here really matter. And so there's a need for the space of training data to sort of cover this entire gap, so you can have platform agnostic methods. But then moreover, it's not just that the platforms differ, it's that everybody's looking at a different tissue with a different marker panel. And sometimes, what looks like a positive stain for CD8 is going to be a different stain for Leeat Keren. And then on top of that, the real information that's present in these multiplex assays is in the correlations amongst these channels, right? And so when you think about how humans label these data, most people don't look at a single channel at a time. They look at a collection of channels because that XYZ things are typically co-expressed. So again, this is data-driven methods development. Very first thing is assemble data. And we got a very talented research technician, one of Caltech's best and brightest undergrads, who worked with us for an entire year. And the main thing that she did was that she went through the entire literature and pulled every single publicly available spatial proteomics data set, standardized it, captured all the available metadata, and put it in a central repository where we have this internal version database for basically every imaging data set that's worth labeling. And with this, we can actually start sort of passing things through Mesmer, creating consensus cell segmentation labels, and then passing them through cell phenotyping labeling workflows. And so I know this contains about 20 different data sets. We've curated a subset of markers around 51 that were gathered from each data set. The existing labels that we've pulled in have about 4 million of varying quality. There's non-trivial amount of label noise. There's some estimate of how many cells are in there overall, but I'm not sure how much I currently believe this. It's probably more. And so just what these figures here show is that the channels, just that the markers that are present, just vary across every data set. There's some that are shared, there's some that are different, and this is the challenge that has to be faced if you want to have a method that's just going to work across all these different data sets. And so for generalization, you can write down some things that you need. You want some normalization. You want the model to have full access to the full information content of the images, and so it should be looking at all the different markers at the same time rather than just looking at one at a time. Ideally, you'd also want these models to be aware somewhat of what a marker is, right? So it should know hey, if I'm looking at CD8, an image of CD8, then I'm actually looking at an image of CD8. If I'm looking at CD4, I'm looking at an image of CD4, et cetera, et cetera. And then also you want access to these human loop frameworks of labeling. So this is a slide that I just couldn't stop myself from adding. So one of the students who works on this came from physics, and so we have a hodgepodge of people in our lab. Some physicists, some chemists, some biologists. And when he started working on this, I was telling him, hey, we've got this project, CD3, CD4, CD blah blah blah. And on his notes, he just wrote down, CDN. Right. And so clearly there's a gap that had to be filled of immunology knowledge. And so what student Rohit did was he took ChatGPT and then fed it Janeway and added it to some of the say language model software engineering software frameworks, and what generated was this Jane, which is a immunology assistant powered by language models. Also has access to the OMAPs, sort of some standardized marker panels created that HubMap has produced for the community. And this creates a resource where if there is some question about some immunology question, you can just ask it, right? So if you're looking at regulatory T cells, you can just ask Jane, hey, what are the markers for regulatory T cells? And it usually produces a response that is fairly truthful. It's a known phenomenon that these language models hallucinate. That's been less of an issue with what Rohit's put together. And so internally in the lab, and then also like across different labs who are in the immunology space at Caltech, this has been a surprisingly useful resource. We're in the midst of putting similar ones together for cell signaling, and then a few other spaces where we're active. But we'll see how useful this is before seeing if it's worth the effort to turn it into a paper. Okay, so how do we solve the generalization problem? Starting points similar to what you've seen before. So start, find cells, crop out areas around cells, include some of the masks so that so the model knows, okay, this is the cell, this is the neighbor cells and whatnot. And so you can take these images, pass them through a convolutional network, and get some vector that sort of summarizes what's in these images. Then what we do is we take the actual text for what that marker is, and turn it into a vector embedding. And there are different ways in which you can create these vector embeddings. You can learn it during this task. You can ask a language model to produce these embeddings for you, variety of different ways, but there's the key is that you convert the channel name into a vector. And this vector, it's in the end, it's just math, right? And so you can take this vector, add it with the vector summarizing your image, and now you have a vector, a summary vector that has both the information content and then also knowledge about what that marker actually is. And then you can feed it into a transformer module that operates across the different markers. And so this is sort of inspired by how humans look at this, look at these data, where you sort of compare across the different marker images. That's quite literally, in a sense, like what this model is doing. And so it's sort of a hybrid between sort of the more classic vision models of convolutional neural networks, and then the language models, the transformers. And so what's nice about this is that now you get these features for each channel now, where it's aware of what's in this channel, but also what's in these other channels. It can use that information to make determinations about, okay, is this particular marker present or not present based on the co-expression? What's the cell type based on the co-expression, and also based on the labels you feed it. And so we actually use this framework for both tasks. So the marker phenotyping, positive marker, negative marker, then also the cell type detection. I know I'm going to go over, but I imagine people might want to see this. Human loop framework. We've sort of refactored some of deep cell label to do human loop labeling. And the way it works is we've modified these networks to be uncertainty aware, which is a non-trivial thing to do for these deep learning models. It's well known that they're overconfident when they're wrong. But we figured out how to mitigate this. And so what we've done is we've made it possible to train lightweight classifiers within the browser that leverage what you get, sort of embeddings that you get out of this hybrid convolutional neural network and vision model, have a lightweight classifier on top. And so what you can just do is just sort of label sort of an iterative process, label some landmark cells, train a uncertainty aware classifier, and then run that on a larger subset of the cells. And when the model is uncertain, you can just zoom in specifically on those cells where it's uncertain, correct them, add them to the training data in this iterative way, sort of plow through different fields of view. And so we have modes of this for cell type classification, and then we also have variants of this for the marker positivity cell phenotyping problem as well. Early, this is ongoing work, but the early outputs is quite promising. So here are predictions on a data set produced by John Hickey, who I think he's moved to Duke by now, but if he's still here, great paper, great data, and we've been using it to pilot some of this work. But you're going to see visually, and then also by the F1 score, that this approach works fairly well. And then on the cell type prediction task, this also works fairly well. We've done some well, I want to say first pass. We're now on our second pass of harmonizing cell type labels across different data sets, which is a non-trivial challenge. And so compromise had to be made. But the accuracy is still quite good. And so the combination of the cell phenotyping positive markers plus a sort of a harmonized, albeit reduced cell type panel. We think there's going to be an effective out-of-the-box solution for a lot of these problems. And not surprisingly, this works quite well when you actually do the rigorous benchmarking. And part of the reason why we get such high F1 scores is because we're not just looking at the individual channels, we're actually integrating the information across channels, accessing those correlations. 2 minutes over, so I'm not going to belabor the point on spot phenotyping, but there is this problem in spatial transcriptomics data sets where you have to go from, I found these spots in the different rounds to what genes they are. This is a hard problem from the data perspective. We've figured out an effective solution by using weakly supervised machine learning, gain consensus among classical algorithms that exist to create higher quality training data where the label noise has been effectively removed. And because this works very well, you can actually pair it with a probabilistic gene decoder and Mesmer so that you can have an end-to-end pipeline for image-based spatial transcriptomics. In goes the images, out comes your spatial gene expression maps. And this is something we're also finishing up and should be public by the end of the week. Okay, so I should stop talking because I've went over, but I have to give a huge thanks to all the different lab members who've worked on these projects. So you saw work from Julie Wang, Rohit Dillip, Laura Prodhen, who I think she's moved on to medical school at Yale, and then also Morgan Schwartz and Ross Bradowski. Huge thanks to collaborators who've been with us over the years as we've been hammering out these problems. And then lastly, I have to give a huge thanks to a variety of different organizations for funding and support. So that I'll stop talking and open the floor for questions.
Thank you, David. So, question for you.
Yeah.
So, back in the day,
Yep.
when we first started looking at all this,
Yeah.
I think, we were sort of maybe in the minority and thinking that these types of problems could not be solved just purely, with unsupervised models with no labeled data.
Yes.
And right. And
yeah.
sort of right. I mean, I'm
Yeah.
sure you remember.
Yeah, I remember. Yeah. Yeah.
And it's sort of one of the themes I think of this session is I think every speaker
Mhm.
has sort of, invoked the strength of having some sort of, human in the loop annotation or
Yeah.
sort of synergizing, these models with sort of,
Yeah.
targeted expert curation. Do you think that's sort of the way it will continue to be? And two,
Do you mean, do I feel vindicated?
huh?
Do I feel vindicated?
Oh, well, that's the second question. So
Yeah.
the second question is do you think that is that sort of is that opinion, is that sort of in the bubble for this session, or do you feel like the field in general has started to appreciate the need for labeled data now?
Yeah. So I think there are people who understand what labeled data can do for you, and then those who don't. And the ones who have come to the understanding, I think that they're making like substantial progress in getting effective solutions out there to the hands of people who are doing these studies. And the ones that don't. So I'm not. I think there's still a lot of work that has to get done, but I do see it more as that, like it's more work now than like a research problem. I think it's like fairly clear that this sort of methodology is can deliver these solutions, and it's just, are people going to have the wherewithal to just do it or not? I will say there's a lot of interest, both academically and then also like commercially. And I'd say like it's debatable in which spheres can make the most progress, right? Because once you're in the commercial sector, you have access to larger amounts of capital and potentially scale. So we'll see, right? It's a But I do think that this is like the sensible path forward. There's always details that have to get hammered out, and there are things that can make the human in the loop labeling. But sorry, yeah, there's always things that can make the methodology better. But as far as what to do, I think this is like the solution. Yeah, and I think that's been borne out. Yeah.
And so what's your prediction? How long till I have just a magical box that will just take my data and classify my cells for me? Top 20 most popular cell types.
Well, I mean, you saw like 3 or 4 methods before I even spoke, right? Some of them produced by your own lab. So I would say, you could probably do that today. I think
no, fully automated magic box. Magic box.
If I had to guess, probably 3 months. Yeah. No, because the hybrid, transformer and convolutional neural networks, they're very effective. The only limiting thing is just data, right? And so we've been labeling data, other folks have been labeling data, and once that data arrives, then you turn the crank and model comes out. And there are things that you can do to improve generalization, so that if there are unknown markers, unknown cell types, you're still close enough to what it's seen already. So yeah, we've it's we're not finished, but I would say that the rate at which we're churning through data is quite impressive, in a combination of that with what other groups are likely to release publicly. I think it'll be likely for it to be on this time scale of 3 to 6 months.
3 to 6 months. You heard it here.
Yeah.
Questions?
Very impressive. I'm relatively new to spatial biology in general.
Mhm.
And when you run bulk transcriptomics or bulk proteomics in tissues, there's often a very poor correlation between the transcript and the actual protein.
Yeah.
So I was wondering, is that also is the same true here in spatial biology in general?
Probably. So I have not we have not run a rigorous comparison between the proteomic signatures and the transcriptomic signatures, provided that you're measuring the same thing. There are likely to be cases where there's high amount of correlation. There are likely to be cases where there's little correlation. I think what also muddy the waters are just the feedback loops that exist. And so you can have a causal link between the two without seeing a strong correlation signature. So yeah.
Thank you.
There's a few questions in the chat. One of these I think is a good one. How does the CNN-based models deal with autofluorescence?
Throw enough data at it. So there are 2 ways you can do. You can subtract out the autofluorescence yourself and make it easier for the model to deal with it, or you can just throw a bunch of data at it, and some of it is autofluorescence but correctly labeled. Others has real signal and also correctly labeled. What we saw with Mesmer is that once you have enough data, it stops mattering whether you've done the autofluorescence subtraction or not.
And then how does the deep cell label human in the loop approach differ from CellPose2's approach of using different base models and training on small subsets?
Yeah, so that is a good question. So it's largely okay. So one, I would say the CellPose approach, human in the loop approach, was inspired by what we originally did with Mesmer. And 2, there's just a difference of philosophy between what the CellPose folks are trying to do versus what we're trying to do. They're of the belief that you can have these a large collection of hobbyist models, each one sort of fine-tuned on each data set, and every person can sort of use their own model. We're of the opinion that there should be one model to rule them all. And if you're trying to do systematic analyses that are sort of at this consortium scale, you can just realize why that's a better approach. The reason why you sort of need to, why and it's not like there's good reasons for advocating for like different different specialist models. One is that people have different different data sets, and it's challenging, as we've experienced, sort of pulling everything into like integrated into the same database. The second is that the reality is that because a lot of this imaging is done two-dimensional cross sections through three-dimensional objects, you actually do have a very real degeneracy in your solution space. And one person's preference for degenerate solution A is going to be different than another person's preference for degenerate solution B. And their approach is like, okay, we'll let people pick which degenerate solution that they want. Mine is a little bit more authoritarian, but it's dictated by like, okay, who's giving us the dollars to do this? And it's the larger consortia where you do need to have a single model that's just going to churn through everything. Yeah, I will say that there are methods that can deal with the uncertainty in the segmentations themselves, and they exist, but they also require additional labels. And I think you also have to think about, okay, how good is good enough? And I think for a lot of folks, things like Mesmer, they can reach like good enough.
And then one question here on the segment anything stuff you showed. But did getting SAM to match the performance of Mesmer involve fine-tuning prompt engineering or both?
Both.
There you go. Then here's another one. I guess I imagine other people probably have this question too. What do you mean by label noise?
Yeah, so if you bring 5 expert annotators to a labeling party, for some sets
most exciting crazy parties there are.
Yeah, for some sets of the data sets that are labeled, there's going to be like a large amount of like consensus, and then for other sets of data, there's going to be disagreements among those labelers. And so there's a degree of noise where it's like reasonable people are going to disagree. There's also a set of noise where the disagreements, or I'd say like there's things that like experts would all agree on are not found in said data. And in the rush, one, this is a hard problem, but then two, also the rush to publication means that oftentimes this is part of the work that receives less than its due attention. And we just see that.
I've never seen this before, David. I don't know what you're talking about.
We've seen it, and it calls into question, okay, which labels can you trust and which ones can you not? We tend to do it by labs that are publishing said data. Oftentimes the labels just aren't even made available. So that's thing one, but that's just reality. And so label noise is a polite way of saying that there are, obvious mistakes in the data sets, and we're just trying to give the models the best chance they can to learn what a positive marker is, what a cell type is, and one of the best ways of doing that is just making sure that the obvious things have good labels.
So quick follow-up question on that. Do you think that the noise that you're encountering, the thing that we always talk about is sort of because we know that if we go in and we look at like stuff that we've done with our, conventional approaches, there's undoubtedly going to be some rate of errors in there.
Yeah.
And and the thing that we always go back to is it's like, well, if it's around, 5-10% or something, and they're reasonably they're not systematically wrong.
Yeah. Kind
of comes out in the wash. So the question I have is, do you find the errors you're finding when you're looking across data sets like this? Do you see that there's systematic biases in the errors, or is it more, and if so, do you think that makes it a harder training task?
So they exist. So like, yeah, so in some cases you can see sort of the systematic errors that happen from just taking like these classical clustering pipelines and then doing some fine tuning, but it's like fine tuning like on with the starting point that has like some amount of bias in it. We see that, and I'm saying like it's not that's actually not the case. That's so bad, just provided you have enough data from where you had enough different clustering algorithms to start so that you don't learn those biases, right? You can sort of average them out. What I'm talking about is just stuff that's just like, it's like wrong, right? And you just gotta, you have to like make sure there isn't stuff that's like obviously wrong that's getting like thrown into the model training pipeline. So yeah, I'm sorry. I'm trying to be polite because I know this thing's being recorded. And I'm up for tenure in a year, a few months. Need some letter writers, and last thing I want is like, well, that asshole Van Valen guy, badmouth our data. No, and I get fired, have to find a new place to live. Just like no. So, thanks for making me be less than polite, but yeah, that is a problem. And so yeah, we've ingested, I'd say like well over 20 different data sets. I want to say it might be closer to 30 now. Many millions of cells. Some of them come with labels, and we know that of those that are labeled, some are good, where we don't think there's any additional work that has to get done. Some are not good, and they just effectively need to be relabeled.
Okay, well, thank you, David.
I hope this works.
Okay, up next, our last talk before lunch is going to be from Albert Tsai, who he's going to do our sort of role of the pathologist session. So I think this is going to be really great. Albert is, I think he's going to have a really good perspective on this because he's a MD PhD by training. So he kind of sits right in the middle of both spheres, and I think can speak to sort of what would be what's most useful for the basic researcher to be thinking about.
All right. It's a little bit nerve-wracking. Here we go. All right. So I'm a pathologist at Stanford Hospital, primarily diagnosing blood and bone marrow diseases, and I'm doing research with multiplexed imaging. I have no conflicts of interest, though I wish I did. All right. So in this talk, I'll be overviewing broadly how I think about histology and diagnosis as they relate to spatial studies. And first, we'll cover a bit about specimen processing, then some general concepts in histology and pathology, and then I'll talk about the importance of histology and protein expression in diagnosing blood diseases in particular, and if we have some time, how we're kind of running the MIBI these days. All right. So the natural place to start is with your question. Most spatial studies compare two or more things. So for example, do differences in cellular composition, arrangement, protein expression, et cetera, explain biological differences or behaviors in normal versus disease, young versus old, progressors versus non-progressors, and so on. And so next you need to get samples from the things you're comparing. And for human samples, pathologists at any large institution can search and pull from millions of FFPE blocks and diagnostic slides. And for a lot of diseases, pathology is just one part of an overall diagnosis. So we use a bunch of like code words. So at least when I was a resident, the report didn't say like celiac disease at the top. It said villus blunting and intraepithelial lymphocytes. So the pathologist is going to help you with all these code words, which you might not know up front. And then pathologists are also going to pick out like clean examples for you. But a lot of entities in the real world have diagnostic uncertainties. And then pathologists are often involved with tissue bank and do the autopsies. And then hematopathologists can get liquid blood, bone marrow, and sometimes disaggregated tissue like a lymph node after flow cytometry. And so all samples have technical limitations. I'll go a little bit into those for FFPE. So the next slide contains some images of human tissue if you're sensitive to that. All right. So when you take a tissue out of the body, the first thing that happens is it stops getting fresh oxygen and the phosphorylations and RNA start to change, and then you cut it open and that cuts the fibers and stuff holding it together, and then it can bulge and tear like this. And then you immerse it in formalin and the proteins and nucleic acids will cross-link and the tissue contracts, but different areas contract at different rates, so it can become like knobbly like this. So the overall point here is that cutting and fixing tissue distorts it, and what you're seeing under the microscope isn't really how it originally looked in the body. And on top of that, fixation isn't uniform, and like you can underfix and overfix, and that can adversely affect any assay. And then formalin only diffuses through at about 1 millimeter an hour, and that decelerates. So that might explain some edge artifacts you might get. All right, and then after the initial fixation, the pathologist or assistant is going to sample specific areas, and which areas, what angles do they cut? A lot of that is determined by AJCC staging criteria. So what is stage? Stage is an assessment of tumor invasion or spread, where T refers to the originating or primary tumor, its size, its depth of invasion. N refers to how many and which lymph node regions are involved, and then M refers to metastases, like seeding to distant sites like lung or liver via the bloodstream. And if you're working with solid tumors, especially, it's worth looking up the prognostic tables, because a lot of the time stage is prognostically more significant than all the protein expression and genetic stuff that we're doing. All right, so let's take something simple like colon. T is determined by how deeply the tumor is invaded through specific layers of tissue. So increasing depth of invasion is worse prognosis and gets you from like TIS down to T4. And the specimen itself looks usually something like this. And after sampling the margins and cutting it open, it looks kind of like this. And like you want to make cuts from surface to deepest edge to look under a microscope at how far the tumor has gone through the layers. And so this explains why you're getting the specific sections that you're looking at. And colon is a really simple example, but a lot of things like larynx are much more complicated because there are a lot more structures that you have to sample. And there are certain tumors that if they see by CT or MRI that they're invading like vessels or prevertebral like fascia, then they're considered unresectable. And so you're not even going to see those samples to begin with. Yeah. All right, so after the tissues have been sampled and then embedded in wax, you cut them onto slides. And it takes a lot of skill to do this really well, because it's really easy to get cutting artifacts like wrinkles and tears. And the tissue itself is at most like a few millimeters thick, but pathologists usually only look at 1 or 2 4-micron sections, which if you do the math translates to a lot less than 1% of the block. And like nobody looks at all the tissue, because that would be like completely insane. Like conservatively 500 slides per block, 20 blocks in a standard breast case, that's 10,000 slides for one case, which is not doable. All right. So now let's look at some tissue. Obviously, I can't teach histology in 10 minutes, but I'm just going to give you like a starter set of concepts. And one approach is to start with the structure-function relationship. Like, how do the structures in the tissue accomplish a particular function? So what are the architectural units, say the layers or shapes, and what's their number, thickness, variation? Then within each architectural unit, what's the cellular composition, the number of different cell types and their proportions? And then within each cell type, what's the range of cell sizes, their chromatin density, and so on. All right, so this is the appearance of a normal lung, and its function is gas exchange. So it has a lot of these alveoli or air sac structures. So schematically, like the pink things are the alveoli, and then they're surrounded by these nets of blood vessels. And so like the walls have to be very, very thin to facilitate gas exchange. So there's like less distance for the gas to go from air into the blood. And then, so aside from like these alveoli, you also have like these scattered blood vessel things. So red blood cells don't contain nuclei. And then when you have a mass of bright pink stuff within like an oval, which is a tube cut in cross section, then you're probably looking at a vessel. All right. So let's look at GI tract. So the esophagus is mainly just a tube that goes to your stomach. So on the top layer, you have this epithelium or surface covering. And as the cells mature from bottom to top, they become larger and more dispersed. Beneath that, you have these noodly extracellular collagen fibers. And you also have these kind of dark pebbles without much cytoplasm. These are lymphocytes. And then again, we have these non-nucleated bright pink cells within tubes, which are red cells within vessels. And then below that, you have a smooth muscle layer. And you can see how it looks slightly different from the collagen layer that's above. And smooth muscle cells are very elongate. They're kind of shaped like baguettes. And so the nuclei are also very elongate. And the pink stuff in this layer is intracellular instead of extracellular, and it's actin and myosin. So this is how you can kind of break down one tissue, and then I'm just going to zoom through the rest. So like in stomach, the job of the stomach is to secrete acid to break down food. And so it has these tubes that are lined with acid-producing cells, and the acid kind of goes up the tubes and towards the food. Then you have the jejunum, which absorbs nutrients that are broken down by the stomach acid. And so it has these finger projections to increase the surface area for absorption. You can see like the different layers of it and their kind of cellular composition, get a feel for that. Then colon resorbs the water that's secreted by the stomach acid. And instead of fingers protruding up, these are actually wells that are going down. And so you can see how in this picture, like jejunum and colon have kind of similar layered structure, but the architecture and the cellular composition within each layer is very different. All right. So what I've showed you so far has been very nicely oriented to demonstrate the different layers of the gut, but most of the time it's actually not like that. And you need to understand these things in 3 dimensions so you can kind of interpret what you're actually looking at. So this is what the stomach looked like. They had these acid secreting tubes, but if you happen to cut the section this way, and now you're looking kind of from the top down, it's going to look like this, which is like all the tubes cut in cross section are now like these little circles, right? Same thing with the jejunum. Like if you cut it this way, now you're looking kind of down the ends of the villi, the ends of the fingers. And so it's not that you have like little islands of cells floating around your gut, like without, anything. It's actually, you're kind of looking at the tips of the fingers. All right. And then in colon, you have these wells, right? And they become circles when you cut in cross section. And then here you have these dense collections of raisins or pebbles, which are lymphoid aggregates, and you can kind of spot them at low power. All right. So for wherever you're studying, it's worth reading a histology textbook. This is the one that a lot of our residents use, and it's also online through a lot of libraries. All right, so now let's go from histology to pathology or disease tissue. And these are the textbooks that we tend to use. At some point, you stop needing like the explainer text, so you just go to the Who Blue books that have like bullet point criteria, and that's kind of the thing on the right. All right, so every tissue has its own specialized pathology. I can't go through everything. So this is like just a starter set of not really rules, but soft signs of disease. And these are all just kind of the flip side of the coin to the histology starter set. So if normal histology has certain shapes and layers, then cancers tend to invade through layers. And then cancers tend to be excess proliferations of one cell type, and they're often arrested at one stage of maturation. And then aggressive tumors tend to have these big nuclei with cleared chromatin and prominent nucleoli, kind of like little eyeballs looking back at you. And they often have a spectrum of kind of messed up cell shapes and sizes. So remember, this is what normal lung look like. It has these very thin walls for gas exchange. And then this is a case of lung adenocarcinoma. So let's just focus on the epithelium. And so instead of the very thin layer of cells, now you have these thick bands, and zooming in, you can really see the cellular atypia. So the nuclei are like these little eyeballs that are kind of looking back at you. I don't know if you can see that one. Yeah, you can see that. Right. And because they have the like the cleared chromatin and the prominent nucleoli. And then the cellular composition is also abnormal because it's all kind of one cell type, like where did the vessels go? And so we can go back here. And it almost looks like the vessels are kind of within the alveoli and forming these little clusters. So that's kind of an abnormal architecture. All right. So now let's look at like normal colon, this thing called a hamartoma. So a hamartoma is composed of normal looking cells, but in a disorganized pattern. So this is a diagnosis made by architecture, not so much by individual cells. And instead of like the nice even crypts in the colon, now you have these like crypts that are kind of different shapes and sizes and oriented in different directions. But there's also the subtle thing to pick up on is that the smooth muscle layer has kind of been pulled up into it, which you can see much more easily at low power. And you're supposed to see these things better at low power than at high power. And so the classic description of this type of disease is it looks kind of like a tree. So here's like on the left, you have like a little tree getting hit by a little wave of colon. And then on the right, you have like 2 trees on a little hill. And this is like a hallmark disease called Peutz-Jeghers syndrome. But like I was saying, this is a diagnosis by architecture, not zooming into high power and looking at individual cells. So there's kind of like a saying in pathology that, high power objective equals low power brain. Yeah. Next, you can see there's a lot of variation even within a single tumor. So this is adenocarcinoma of the duodenal ampulla. And at low power, you can see at least kind of 4 different architectural patterns. And you can get a very different impression of this thing depending on where and how much you sample. So this is another place where the pathologist can really help you with finding the areas damage. So you think a little bit about the nature of architecture and what's actually telling us. Like if every tumor was just a bag of oncogenes and tumor suppressors, like a lot of studies kind of treat them as, then every tumor just grows as a ball, right? So there are some tumors that kind of look like that, like this is Merkel cell carcinoma. But if you have something that's like epithelial, like a layer sitting on top of some stroma, now let's just add one cell, which is like a 16% difference, and make the stroma flexible. And so now just by adding that one cell, you've introduced a curve into it. And so there are some tumors like this is, I think, pancreatic IPMN. And then there are other tumors, let's say, that like, I'm going to introduce more cells, but I'm going to make the stroma inflexible. And so now that's causing the cells to squeeze together. And so let's do that some more. And then there are some tumors that look like this, like a tubular adenoma. And then now let's say that we're going to take these cells, but we're going to remove the intercellular crosslinks that normally keep them together. And then that can produce this effect called hobnailing. So the point here is that kind of like individual cells and small areas of cells can just have like slight morphologic changes, but like what we're seeing in architecture is the accumulated effects of these small differences magnified over thousands of cells. So you can spot an overall abnormality at low power rather than kind of rastering through one cell at a time. All right, then there's some analytical considerations for kind of spatial studies that I think are worth thinking about. So like if you take like a layer of cells that's like, these are all the same cell, and then we're going to cut it, right? What you're going to get is a lot of different cross sections. And then if you want to treat histology like cytometry, you have to like try to get back to whole cell signal this way, right? But that requires a lot of assumptions about scaling and stuff like that, which might not necessarily hold true if your cells are different sizes and shapes. And then there are some cells that are just not segmentable at all. Like Shaq has like a 5-foot nerve from his spinal cord up to his toes, right? So you're not going to segment that or represent that single cell in any realistic way. And then there are some diseases that have like cells inside other cells. So I don't know what you do about that. And then, yeah, so, that happens. Another thing to think about is when you're trying to do like multi-omic studies, and like, let's say you're going to make 3 cuts and you get 2 layers, and you're going to do like, RNA on the top layer and protein on the bottom layer. And you can say, okay, well, I'm going to line those up and I get, these things lined up. But like, if you have 2 cells that are sitting on top of one another, then how are you going to tell that like one thing was from one cell and, the other modality was from actually a different cell? And a lot of cells are just not going to line up. So, it's going to be kind of tricky, even if you have like, even if your histologist was like perfect, which a lot of the time they're not. Yeah. All right, so I've kind of gone through a bit about how pathologists look at histology, but thinking about how does that fit in with the overall diagnosis that we kind of do in a real kind of practical sense. And I think here, like hematopathology really kind of leads the way, in that we've been integrating histology, protein expression, and genetics longer than most other subspecialties. And I think it's very different from a lot of researchers, what a lot of researchers tend to think. So when we get a case, we start with morphology. That gives us a range of possible diagnoses. And then we work those up with antibodies, either by IHC or flow or both. And out of like thousands of possible diagnoses, now we've kind of gotten down to one diagnosis or a few related diagnoses. And aside from some kind of specific situations, we tend to get the genetics like weeks later. And most of the time it either kind of confirms what we already thought, or of the 3 things we narrowed it down to, it tells us which one of those it was. And also refines the subtyping and the prognostication. So let me give you an example. This is a needle biopsy with like a lot of blood, and then you have a tiny fragment of cells here. I thought it kind of looked like follicular lymphoma. So these were the stains that we got. It's all 1-plex IHC. There's no like co-registering individual cells from section to section. But we kind of say that, well, most of the cells, or almost all the cells, are expressing BCL2, almost all of them are expressing CD10, and almost all of them are expressing and PAX5. And so like by co-localization and noticing that they have similar morphology, we infer that this is like one tumor co-expressing all 3 of these, and that pattern is good for follicular lymphoma. And for various reasons, they rebiopsied, and weeks later, genetics came back with a BCL2 heavy chain rearrangement, which confirmed the diagnosis. All right, so there's some people who say that we're just going to sequence everything, and that's going to give us our diagnosis. But like we've been doing clinical NGS for a while now, and we found that genetics isn't really specific and can be like horribly misleading sometimes. So for example, BCR-ABL is the first translocation ever discovered. You can see it in CML chronic phase, accelerated phase, blast phase, AML, BALL, MPAL, and rarely TALL. And these are all very different diagnoses with very different treatments. And then what distinguishes like MDS from AML myelodysplasia related, it's not sequencing, it's really just 2 eyeballs in a brain microscopy. And sometimes antibodies can be helpful. CML, also T-cell lymphomas can have some of the same mutations, so that's also another problem. And CMML versus AML M4, M5, that's also classic 2 eyeballs on a brain, and even like antibodies don't always help you here. And then follicular lymphoma versus large cell transformation is also a classic kind of 2 eyeballs on a brain thing. All right, and then there's also some people who say we're just gonna like AI a bunch of H&Es, and like there are some specialties where like that might kind of work because there's some where the diagnosis is almost made entirely on H&E, but in HemePath, we've kind of done this experiment and we know what the answer is. So in the late 1900s, when I was growing up, like all we had was H&E. And like they couldn't figure out a B cell from a T cell, right? So like the lymphoma classification, just lymphoma, not leukemias, all they could figure out was 14 things. And today it's almost 100 things. And in red, I've highlighted the things that are primarily made by morphology and immunophenotype. So all of this is to say that modern hematopoietic diagnosis is multimodal. And most of the things are kind of headed this way if they're not there already. And I would argue that a lot of pathology is kind of barely out of 1982 compared to HemePath. And the way to get to 2022 is to appreciate that diagnosis is not a solved problem. And diseases get discovered or subtyped when what you thought was one disease is actually multiple diseases that have different biological behaviors or respond differently to new therapies. All right, wow, I'm fast. All right, so since we have a little bit of time, just a little bit about kind of how we're running them maybe these days. So if you have maybe you're familiar with this interface where it's like very difficult to set up like more than 5 FOVs at a time. So what we're doing now is we're kind of rastering over like the whole area of a slide with the SCD image, and then we kind of stitch this whole thing together into like one entire SCD image. And then we throw that into an interface where it's all like WYSIWYG, drag and click and stuff like that and draw. And so then it becomes very quick to set up your TMAs. And then we also have like polygon draw. Yeah, so this is fun. Yeah. Yeah. All right. Yeah. So if you're interested in this, maybe you're interested in this, yeah, we can help with that. All right. And yeah, that's whoops. And yeah, that's it.
Albert, that was great. So the question I have for you is, you were showing a second ago, was it a colon carcinoma case where you were showing these different compartments of cell morphologies?
Yeah. Mm-hmm.
And so, and you mentioned briefly in there too, sort of the vast amounts of tissue that are either surgically resected and processed versus what actually gets analyzed, and I think this is kind of a point that very well, which is that there's all kinds of heuristics and sort of predetermined guides for how this stuff is accessioned, which is kind of biasing what we're cutting out, biasing what we're looking at, and even though it's and we're still looking at only like a subset of a lot of stuff a lot of times, which I think is maybe not like completely known outside of pathology circles. And so with that in mind, like what do you think about when someone's coming into this that wants to study, say, PDAC or breast cancer, and they're not familiar with all this? Like what should they be thinking about? Like, should they just be working with the pathologist? Should they, how does someone that's coming in that doesn't know all of this, what are the things that they should know that they might not know?
Yeah, so for somebody who's kind of coming into this like without a lot of background knowledge, I think that like getting your pathologist involved early is going to help you kind of do a lot of like simplification, because they're just going to explain to you with all the code words and all the stuff, filtering out like down to what is kind of essential for your study. And like for you to learn it on your own, like you can start with like a histology textbook and then look at like a pathology textbook. But like if you're going into it without knowing all the nuanced details, then it's going to be a slog to kind of do that. And then like to get the samples to begin with, you're probably going to need a pathologist to select them out for you.
Any questions from the audience?
I have one, Mike. That was really interesting. Underlying a lot of what you said was the concept of scale and how at certain scales things are very obvious and others they're not. And I wonder if there's some sort of a rule of thumb that you have, come up with, like when you have to drill down a little bit further, or when you're pretty confident at a certain scale of what you're seeing, and just that whole idea. If you want to talk a little bit more about that, I'd love to hear it.
Yeah, so that's a great question. So every specialty has its own kind of sets of structures and scale. So like in hematopathology, a lot of the stuff we're looking at is like floating single cells. And so we're going down like higher power than in a lot of like solid tumor pathology where they're, 10xing a lot of that stuff. And so like, I'm routinely like 20xing.
You mean the objective and not single cell
the
sequencing?
The lower objective a lot of time is, for me, it's for a search path, it was like 4 or 10x. In hematopathology, it's like 20, sometimes 40x. If you're a resident, it's like 100x oil. Yeah. But it depends on like specific each particular tissue type. Like the architecture is going to be specific to that particular type. Yeah.
Yeah, I was gonna. You have fans. There's one comment here that simply says, "I love you, Albert."
Thank you.
And so you're the only one. You're the only one. So, here's one. How do you QC your slide in order to be confident that your slides are fit for a multiplexed imaging study?
That's a good question. How do I think about that? So, like, for initial first pass, you're going to get an H&E on like every slide that you get, and like you can usually see on that H&E whether there are a lot of really obvious kind of artifacts that make the cells look, not right. And I guess if you start doing just like a couple of IHCs, you can often kind of figure out if you're not getting the reactivity you're supposed to be getting. So at least for what we're doing with kind of HubMap, we have like an on-slide array control that's going to be a set of positives and negatives to go along with our sample tissue. And so if the QC tissue that's the same on slide, that's also on the slide is not staining properly, then we're going to probably flag that for, not including that data.
Hi, yeah, great talk. Super informative for me. Haven't been around pathology too much, but how much as support do you provide to people for clone selection, for instance, when they want to think about moving some of their assays maybe to pre-translational or preclinical stage?
Yeah, so that's a good question. Like, so if I understood it correctly, how much does pathology help with bringing up assays? Is that kind of your question?
Yeah, a little bit. And maybe do you have to advise on that a lot? I mean, what's the interaction like between you and the community here, for instance?
Yeah. So it's not going to sound like great, but it's usually a lot of like personal, like emailing, and like if a pathologist is interested in your topic, then they might help you out. But if they're not, you're going to have to hunt around maybe. So like at least with like certain departments at different institutions are going to be like, the highlight, department or something, and they're going to get more people, more funding. And so like those are areas, the places where you might get kind of more support because there'll be more kind of people around to help you with that. Other kind of specialties where it's like, we've had some specialties before that we're all down to like one guy. And if that guy went on vacation, like everybody else would be like, in a lot of trouble. So I think it really kind of depends on like what the structure of your department is and who's kind of around to really help you with the different types of studies. Sorry, it's just kind of how it seems to be.
Then another one here, kind of similar lines to the question I was asking before, is essentially it's asking what kind of histochemical artifacts like tissue artifacts do you sort of keep a lookout for to, just on like H&E and stuff like that?
Yeah, that's a good question. So the artifacts on, we'll be covering a couple of them actually in the histology session later. So there's like giant lists of artifacts that you can get for IHC specifically. Like you can get edge artifacts like on the edges of tissue, maybe because like there's differential fixation, those areas can be slightly different. Like necrotic areas can bind antibodies non-specifically, and so you get a lot of false signal where you get necrosis. And just there are a lot of crappy antibody clones out there, like that just do things that you don't like expect, like like KS67 in the neutrophils or something is something that you seem to see a lot, but the neutrophils are not supposed to be mitotically active. So things that just don't make sense. And like, I think that this is kind of where your pathologist can really help, is with your positive negative controls to act as your kind of QC.
And what are your thoughts on optimal antigen retrieval solutions?
Is this from Sean?
I know. It could be. It says anonymous.
Right. Okay. So clinical IHC is all optimized. It's one or two plex, but mostly one plex. And so like we have like, I pulled the list of everything that we have, and most of our stuff is pH9 retrieval. Some of it's like protein retrieval. Some of it's pH6 retrieval. The theory has been that pH9 is like, it retrieves better, but like pH6 has kind of lower background. But those aren't universal rules. You really just have to kind of test it out for yourself. And what I like to start with the clinical clones that people have validated for decades. So there's a site called NordiQC where they have like all the clinical clones that people have used, and they've like formally validated them in a clinical lab. So I usually like to start there. And yeah, so I hope that answers that question.
I have one question, if we have time.
Yeah.
Okay. Thank you so much for such a great talk, especially for me who doesn't have a basic, I have some basic pathology knowledge, but not as much as you have, the deep knowledge about the histology and all this stuff. So my question is that you mentioned that co-registration is not always perfect. That's one of the comments that you mentioned. And then multimodal imaging will help this, this problem. So the thing is that I was thinking that if PET imaging or MRA or MRV as a, just as a different modality can be helpful to answer this question that because yes, co-registration is not always perfect, but if we add this information, then we would be able to, maybe in disease cases, for example, for unknown, tissues in rare disease, for example, known disease, we are good, but for rare disease or unknown disease, we need to have such a collaboration with these two modalities. So what's your thought? And if it is possible, how do you see the future of this idea?
Okay, yeah. So I guess the question, is there a way to like improve co-registration by using like additional modalities or something like that?
That's correct. And especially MRV and MRA and PET imaging.
Okay, yeah, I don't know as much about those in particular, but so what I was referring to in terms of like co-registration being tricky is when you have multiple sections. So you have like a pink section on the top and a green section on the bottom, and you're trying to align them to each other. Like if you have like let's say a multiplex imager, then theoretically at least, like the co-registration for that modality should be perfect within that modality because it's multiple things in one layer, right? But like I was thinking more about this instance where you have two different modalities on serial sections, and so the two sections are going to be very slightly different, like because the plane of sectioning or the thickness of the sections are kind of too large to be, you know, to get like an individual cell, like a smaller cell, like all in one section, then this is kind of what I was referring to in terms of that. In terms of like kind of, I don't know that much about kind of the MRV that you're
Magnetic
referring to.
resonance venography or
Venography.
magnetic
So that would be in,
angiography.
that would
So
be
they are
like,
all blood vessels. So
oh, okay, so that would be in like a living patient?
yeah, inpatient. Yeah.
Okay,
It's
okay,
live
so
imaging, you know. So
yeah, yeah. So especially like I've been thinking some about this for other things. Like when you have like one patient, right? Like it's one kind of quote unquote sample material, and then you're going to image them with one modality, and then you're going to image them with another modality, and then you can kind of line those things up. And especially like PET CT, I don't know if you've seen PET CT, where you have like a high resolution CT and a low resolution PET, and then like because when you put them together, you get more information than kind of either modality alone. For tissue blocks, because your assays have to be done on different like physical sections of tissue, I don't know that that kind of perfect analogy applies as much, because it's like because when you're doing like whole body imaging, it's the same thing, whereas like with tissue sections, it's different like things. So even though they're pretty close, they're not going to line up with tissue sections. I don't know if that answers your question.
no, my point is that when we have the MRV, for example, images, and we have the tissue, then from what we have obtained from our MRV, for example, or MRA, then we can co-register. Not at the same time, absolutely, it is not possible, you know. But because they have the same concept, like it's blood, maybe we can get the information that we need, especially for complicated, you know, disease. But I just don't know.
I think I'm just not like familiar with exactly kind of what your application is, so I can't speak to it when I don't really know. But we could talk later.
Yes, of course.
Yeah. I think you'll have to explain to me kind of what the technologies are. Yeah.
Okay, there's another question here about formalin fixation, which is, you know, how many hours of formalin fixation do you apply for tissue samples? How much does variable amount of fixation affect multiplexed imaging?
Oh, that's a good question. So what kind of variation is there in fixation? So like a lot of the fixation is done on the surgical schedule and not so much controlled by the pathologist a lot of times. So like a lot of tissues, you dunk them in formalin, and if it's like 6 o'clock, everybody's going home, they'll sit overnight. And then usually like when you do like an initial fixation, that's to just kind of harden up the tissue a little bit, so that way you can cut it. Otherwise, you know, if you're trying to cut raw tissue, it's kind of like a mess, right? So like after initial fixation, which is usually how many hours to harden up the tissue, then you're going to cut it again into finer slices. And when you cut it into finer slices, like the individual slices should be no more than like 3 millimeters thick or so. And so that way the formalin diffuses in from both sides, and then you can get through like 1 millimeter per hour theoretically. So you should take more, like, you know, at least like 3 or 4 hours for that little thin piece. And like a lot of the time it's sitting there for like 8 hours, because it's going to sit in formalin until it gets to the tissue processor a lot of time. So the exact like fixation times like are going to be variable depending on like surgical schedules and kind of exactly when the things were taken out. And like so there's a lot of variation in that. You can try to kind of limit that if you like are able to get the specimens fresh out of the OR. And then in terms of like people have done a lot of studies, I think, with fixation, and I think the consensus was that like fixation beyond like 24 hours or so, like it wasn't terrible, but once you start to get too far beyond that, then it can get worse. But like under fixation is also a big problem, and like under fixation was more of a problem than over fixation. but like I haven't looked at those particular studies in a while. I think it's worth probably for whatever tissue you're looking at to see if somebody's actually done those studies.
Yeah, if for anybody that's curious, David Rim at Yale, they've looked at this a lot. I had a conversation with him a few years ago, and he kind of reiterated what Albert's saying, which is that probably the biggest cause of artifact is not usually fixing for too long in formalin. It's the tissue sitting ischemic and not getting fixed. So the ischemia time is like by far seems to be the biggest source of artifact. And unfortunately, it's only, correct me, is this right, Albert? It's only breast cancer cases where there is a requirement on when the tissue is getting processed of how long the tissue is allowed to be ischemic. Otherwise, it's like, sort of, is that right? And
Yeah. So a lot of the studies were being done like towards the end of my residency, and then I went into hemepath where we don't worry about any of this stuff. So like that's kind of what I recall, is that people were doing formal evaluations of formalin fixation because of the breast ERPR studies. But for other tissues, I don't know if they've been done. I'm not up to date on that.
Yeah. So, and this, at least the last time I looked at it, one of the secrets, not flattering secrets of like, pathology, is that there is like essentially, with the exception of breast cancer cases, there's no mandate about how long tissue can sit before it's fixed and processed. So it's been implemented in breast cancer because they found that it impacts like ER staining and stuff like that, but in other cases it hasn't been in most places. One last question, Albert. When you're putting together a study for some of these, for some specific cancer, what do you think about in terms of how many patients should go into that study? How many different areas that you want to sample to image? What are kind of the general categories about what you would think about? Let's say like for let's say it was colon cancer. Like what are like the general things that you would be thinking about for what you would think for getting a good representative sample of a patient, and how many patients you think you would need to kind of get an idea of what a specific, subgroup looks like?
Yeah, that's an interesting question. So I don't have like a great answer, but like, when I'm kind of approaching a lot of this stuff, is I just kind of look at it on H&E so I can see differences by eyeballs. And then kind of, how many do I really think that there's like a difference? So like, let's say that I have two different biological behaviors. I have like, let's say disease progressors and disease non-progressors. So like, somebody with DCIS, half of them progress to invasive tumors, half of them don't, let's say. Like, can I see a difference, by eye between these two different groups, and then what are the ends behind that? How many do I need to evaluate to see that? But beyond kind of like what I can see by H&E, like, thinking about what are the other kinds of studies that have been previously done on these, like what kind of RNA studies have been done? Have people found other things that correlate with these biological behaviors, like to provide some kind of end behind how many samples to take? But that's tough. Yeah.
Well, thank you.
Thanks.
So we're about to have lunch, and I think at 1 p.m., is that correct? Albert will be having his workshop where within 1 hour, you will know all of histology and how to look at H&Es. So where's that going to be? It's going to be here? Okay, in this room. Thanks.